thought you could use something as simple and as common as a thermometer to determine whether you are at risk of a heart attack. And this is exactly what we've done in my lab. We've combined a simple thermometer and functionalized it with polymer particles. And these polymer particles ensure that it's very sensitive for biomarkers that are released when you have damage to your heart. And if you want to read more about it, you can have a look at the paper. There were a couple of challenges along the way. And the first thing you have to imagine is, how do you actually get these polymers to stick onto a thermometer? And that's not as easy as it seems, because most of the polymers we used to work with are powders. So the powders itself, they just fall off from the surface and it's not stable. We try to prevent this by using a type of glue, where we kind of glue the polymer particles onto the thermometer. And while this does work and it would stay in place, you can't get a coverage of more than 20%. Now, the way we circumvented this within this paper is going to smaller particles. So we moved from something which is micro to nano, which is 1,000 times smaller. And if you think about what a nanoscale is, your hair is about 100,000 nanometer. So what we were working with is around 100 times smaller than a strand of your hair. So basically, you're not able to see it. It does make it a lot easier to work with. So rather than having to put the powder on the surface, all we did is we just applied a drop of the water with the nanoparticles, and it stuck beautifully to the thermometer. That's one problem solved. The polymers are actually on the thermometer. But now the key thing is, why do you see changes in temperature when you get binding to this polymer layer? And for this we have to look in a little bit more detail. And what you can see here is that these polymers are actually porous material, so they basically got holes in them. So as soon as something binds to these holes, you're basically adding another resistive layer, so you're increasing the overall resistance. So as a thermometer is functionalized with these polymer particles, here you can see that when you get binding within these cavities, the temperature will go down, because again you form an additional resistive layer around the thermometer itself. But actually this platform is really versatile. By adapting the polymer layer, we can look at other important biomarkers, such as those which indicate when you are at high risk of cancer. So we studied these cardiac biomarker concentrations not just in buffered solution, but also in real serum samples, which can be difficult to study, because within blood you've got a lot of other proteins which might interfere with the signal. So we showed that our sensor was very sensitive, very selective, and very adaptable. What are we going to do with this in the future? We're going to explore a number of options. So for instance, we're going to look at patient samples, so real clinical samples, and compare our technique versus already existing techniques. Because the idea is that the sensor platform that we have, it's low cost and it's very fast, so you should be able to get results within a couple of minutes. Within this playlist, I'm going to summarize more of our research papers. So if you do want to stay tuned for this, please subscribe to our channel.